So our next speaker today is Barry Ryan from Biomedical AI CDT in University of Edinburgh. Today he'll be talking about an integrative network approach for longitudinal stratification in Parkinson's disease.
Actually, a few days ago.
Yes. Oh, it's over here.
Sequential model, it's different.
Could I get the lapel mic on you, please? Sorry. Thank you. All right, here you go. Okay, whenever you're ready, take it away.
Okay, nice. Yeah, so my name is Barry Ryan. I'm a PhD student from the University of Edinburgh on the Biomedical AI CDT cohort. So yeah, thanks very much for having me as well. It's kind of a fun opportunity to, I guess, talk to a more ML audience coming from the Biomedical AI cohort. Often it can be very biology associated, so try and stick with me for the next half hour and we'll make it through it together. And also, if you have any questions, just throw up a hand. I'm very happy to kind of take it during the talk. Yeah, so I'm going to talk about an integrative network approach for longitudinal stratification in Parkinson's disease. And other than Parkinson's disease, what does that mean? So I think starting on the application is probably the easiest way to do it. It is quite a recognizable disease. It is the second most prevalent neurological disease, particularly in older people. So it is an important problem to tackle. And how we're doing that is we're taking a so-called integrative approach. That means that we're going to be doing some multimodal analysis. We're going to be trying to take in different sources of information for that. And the way in which I've, I guess, done this methodology is through the use of networks. So network biology is all the rage at the moment. And thankfully, I've come in time just for my PhD. And what's different, I guess, about this, I suppose, methodology and the research is that you kind of get to do this longitudinal stratification. That means we're going to be applying this method at different time points of the Parkinson's disease in these patients which we're looking at, and hopefully be able to track that in some way. And stratification, essentially, we're going to have 3 different stratifications. So we're going to have healthy controls, we're going to have individuals who have Parkinson's disease, and then we're going to have some people in the middle. And I'm going to call those prodromal individuals. So prodromal is just another word for someone who hasn't yet reached a clinical diagnosis for Parkinson's disease. And that whole approach has kind of been the culmination of my PhD focus for the last couple of years, and a methodology which I've called multi-omic graph diagnosis, or MOGDX for short. And finally, I'm going to give the results or the outcomes for this work so far. So why is Parkinson's disease interesting, and also why is it really difficult? So really I guess if you're to Google Parkinson's disease, the first thing that you might see is that it is as a result of the death of dopamine cells in the brain. We've been able to account for this death of dopamine cells due to some genetic associations, due to much research at people who are far more intelligent than me and far more better at biology than I am. So I'm going to, take those genetic associations for their word on that. And that can account for about 30% of the disease so far. So like you'll see studies anywhere between 25 to 40%. People will kind of link that with genetic associations. So what's going on with the other 60%? And even in those genetic associations, why will some people have this mutation or this kind of change in their DNA, but not have the disease so far? So there's lots of this kind of like complexity, heterogeneity, and differences in individuals with the disease or without the disease or in early stages of the disease. Particularly, this can kind of come about in terms of differences in their symptoms. We would always associate kind of these motor symptoms, shaking with Parkinson's disease, but also some people will have smell disorders, some people have trouble with their sleep. People can take different medications. This medication can vary individual per individual. So quite likely it is that you'd have 2 people with Parkinson's disease. Medication might be effective for one for 5-10 years and they'll have really good outcomes, and for the other person might not be effective at all. So why is this the case? And how can, I guess, we untangle all of this heterogeneity and differences in different people? And the way in which I'm going to motivate and the way in which I've kind of focused is genomic measures. So we're trying to explain this kind of, I guess, really broad differences in individuals by lumping them together and being able to find patterns in their genomic profiles. And the data set in which I've done this analysis on is the Parkinson's Progression Marker Initiative, the PPMI data set. I would highly recommend checking it out. It is fantastic curation by the actor Michael J. Fox, who's funded a lot of it, and they have over 900 Parkinson's disease patients. I've focused on genomics, but they also have image scans. They might even have voice audio signals as well and everything. So it is great. But for the focus of this research, I've done genomics. And the really interesting thing here is that we're working with whole blood samples over kind of this 3-year time period in a lot of Parkinson's disease patients. And why is that important? Well, Parkinson's disease is a neurological disease. So it's happening in the brain. So if we can find signatures or signatures for this in the blood, that would be really, really interesting, because you have this thing called the blood-brain barrier, which means lots of your features, which would obviously be very informative in the brain, may not be circulating or may not be present in the blood. So can we actually find a signal or tease a signal out for that? Also complementing this with CSF, which is important to say that that is not a blood sample. That is a plasma protein, which you can obtain. So you'd actually expect a much stronger signal for the brain in this plasma protein. And why we want to do this, I guess, in a integrative or multi-omic fashion is because these measures, yes, they're all very interrelated. You will find common shared signals before that. That is a good thing. That is what we want. But I mean, for the people who've been doing this a lot longer than I have, no one's been able to figure it out with just one of these so far. So can we actually get a much bigger picture by combining these? It's like joining different pieces of a puzzle together. Can we get a much more clear image on that? And yeah, this is quite a bit to take. So take a little step back, and really, if you understand nothing else from this, and I'll be going to more detail, it is that we have these modalities. So these are going to be our genomic measures. And at the very end, we're going to get a patient classification. So that is going to be our stratification of healthy control, Parkinson's disease, or prodromal, which is just early indicators for that. So the methodology here is that essentially we're going to use each of our genomic or our omic measures twice. We're going to represent each omic measure as a biological network, specifically as a patient similarity network, and I'll motivate why we do that later. And also, we're going to do some kind of, I guess, taking these genomic measures as they are, and also getting, I guess, a kind of integrated genomic profile specifically. And we're going to combine both the network science with kind of more typical ML, smaller, lower dimension representation of that. Whew. Okay. So, patient similarity network. What is a network? A network is a node, an edge, and a group of that. What is a patient similarity network? It is simply just that we have patients as our nodes, and we're going to measure similarity between them. It is as simple as that, as taking a correlation or a distance matrix, depending on what you want to measure. Why would we use a patient similarity network? It kind of is analogous to medical practice. If you go into your doctor tomorrow and you're saying, I have a sore throat, and I have this symptom and that symptom, they're going to say, okay, well, I've seen this before. And you're like, thank God, they've seen it before. It's not just me. So the same kind of, I guess, knowledge here is that people who have similar profiles in a specific modality are going to have similar disease classifications and outcomes. And I think a really nice quick example of this is that you can actually tell a story pretty quickly with a network. These are going to be people who have a genetic association or a mutation in a gene for Parkinson's disease in orange, and you're going to have the blue with healthy controls. It doesn't necessarily mean that everyone in orange has Parkinson's disease, and it doesn't mean that everyone in blue doesn't have Parkinson's disease, but you can extract information from that really quickly. So stepping back out now to once we have created this similarity network for each of our genomic measures, how can we combine them? And we do that through another algorithm called similarity network fusion. And again, I'm just going to show kind of this DNA methylation, which is another genomic measure in which we use, and how we get a different story. Here you can actually see that much more clearly. You might have this group here who actually have a clinical diagnosis for Parkinson's disease, and those who are healthy control. So you get that kind of different omic measures are telling different stories, and we want to be able to fuse these together and combine these together. So why we like similarity network fusion to combine our modalities is because it's just really, really good. I would highly recommend it, particularly for low numbers of modalities. You also get this really nice kind of retention where you get to retain patients which are missing nodes. And it's really good at sharing information across modalities, particularly this kind of Game of Thrones ideology that a friend of a friend is a friend, or an enemy of an enemy is a friend, or whatever that one is. But it's really good at sharing information across each other. So it's kind of boosting these edge weights where it's uncertain about what the connections are in the network. So stepping back again, what we've done is we have our genomic measures. We've created our network. We fuse this network into one single network. So now that we have this one single network, we want to be able to learn from that. and we're using a graph neural network for that. But for the graph neural network, we need to have a node feature joint in that. So we want to learn from the network, but to do that, we need the node feature. So how do we get the node feature? So that is kind of this, I guess, genomic measure or profile, which I was discussing about. So we go back to our original genomic measures, and we're going to integrate them again in another way so that we have one lower dimensional representation per patient. So why do we need a lower dimensional representation? It's because biological data sets are really tricky and they're really, really big. They can be really annoying. So for example, DNA methylation might have 800,000 features, and you might have proteins which might have 2,000 features. So if you were to just concatenate them or just, naively put them together, your DNA methylation is going to completely overwhelm and overpower the proteins. So we need to be able to reduce the dimensionality, so that's kind of a fair fight, so that the big guy isn't always winning. And the way we do that is this multimodal encoder architecture. So we basically do a 2 layers. And the reason for 2 layers is because we want to capture interactions between features within each omic measure, as well as just the raw features themselves. We can press it down, we do our imputation. Again, that's really important because we don't want to be losing patients. We don't want to have to intersect between our different omic measures for each patient. And then, just typical ML AI in which we kind of pull these layers together, and then we want to perform node classification with a graph convolutional network. And what's a little bit different here is that we've extended the training back through the graph convolutional network so that we're training both this encoder architecture and the graph convolutional network concurrently. It means that our propagation is going the way through so that we're actually reaching the raw inputs from the omic measures. So if that completely lost you, the only thing that really matters here is genomic measures in, patient stratification, patient classification out. Cool. So back to Parkinson's disease. How does this all fit together and how does this all work? So the measures which we have are these different gene expression measures. That's your mRNA and microRNA. We have DNA methylation, we have SNP genotype, we have CSF plasma, and also we have clinical health records. And we have these different groups or samples. So we have our healthy controls, we have those individuals who have a mutation in a gene and also have Parkinson's. Then we also have individuals who have Parkinson's disease but don't have a mutation in the gene. We also have those prodromal who may or may not have a mutation as well. So it's basically just this big bag of mess. And essentially what we've done is we've just tried and brute forced every single outcome that we can. It's the great thing about university resources. They don't really limit you when you just want to try everything. So what I've done is taken every single combination of modalities that I can. I've done it individually, and I've done all the joint, and I've done it for kind of healthy control versus genetic, for healthy control versus idiopathic, and for everything. And I think at this point, it's really important to acknowledge why you do something, particularly in a biomedical field, because as I have up there, telling someone with Parkinson's disease that they have Parkinson's disease isn't exactly helpful. They're not going to thank you for that. So why is this useful, and what is nice about this? And at its very fundamental, often you'll get biologists and PIs and they'll be like, I've done this massive study and I've created 5 different data sets and it's going to be the most amazing thing ever. And it's, well, if 4 of those data sets aren't actually informative, you're wasting a lot of university money there. So it's being able to narrow down maybe perhaps what information is important, finding the features which are most selective or informative of that disease. So what are the disease signatures in blood samples? Is there subpopulations? So instead of having to, for example, do 1,000 individuals, what if you just identify 200 individuals who are going to be much more representative of the problem that you're trying to solve? Things like that. That's the, I guess, difference between many analysis, is that this may not necessarily solve Parkinson's disease, but it might be an initial step to be able to get more information to inform it down the line. And then these things can lead further into early disease detection, disease stages, inferring trajectories, and what other subtypes might be able to inform a clinical trial, for example. So after running every single possible combination of modalities, these are the best ones at each time point for the different subgroups. So if you remember, you've genetic and idiopathic, so those are the groups. That means when you join together those who have a mutation and those who don't, genetic is just looking at those who have a mutation, and idiopathic is just looking at those who don't. And again, we're stratifying whether they are a control, have early signals for it, prodromal, or actually have the disease in which itself. And I think the first thing to note here, and if I had time I was going to draw boxes around all these, but what kind of comes out is that DNA methylation seems to have some sort of shared signal across all of these groups. So that one single modality kept coming up again and again and again as being informative. So we can kind of conclude that, okay, there must be something there with the DNA methylation which might be capturing some sort of disease signal. And the next kind of thing to look for is that when you group together genetic and idiopathic, your accuracy is a bit rubbish, which means essentially what you're doing is when you're kind of coming together with these two different groups is you're getting a lot of crosstalk, which isn't necessarily a good thing. But again, it tells a story in itself in that, you know, maybe there's a reason why these idiopathic individuals are so much harder to pin down what's causing their diseases, because it is actually quite different to all these genetic associations which we found for Parkinson's disease. So perhaps there's something else going on there, and you know, Parkinson's disease is this big umbrella term which actually might encompass different ones. Potentially that's a kind of another clue. What is really nice is when you focus just on the genetic individuals, so those who have a mutation in a gene for Parkinson's disease, your accuracy kind of suddenly explodes. And again, that kind of makes sense, particularly when you think of it biologically. We are using genetic biological measures, genetic biological features to predict something that is found to be genetic, right? So it's nice to put up good accuracies, but inherently, if you think about it for a second, it actually makes quite a lot of sense. What is interesting here is that, for example, we can see that there's this really clear signal in these single two modalities or omic measures which we found. So again, looking in more depth into that, what story does that tell? And then finally, if you just look at idiopathic, again, it kind of is a bit more of a mess, which again is reflective of what is probably happening in that disease, is that we just don't know what is there. Is there a genetic signal there at all? What's coming out is that CSF is quite important, which again probably makes sense, particularly later on in the disease course, because CSF will track the alpha-synuclein, or will be a measure of alpha-synuclein, which for those who know about Parkinson's disease essentially increases as the disease progresses. So looking again, these plots basically are showing, when we group everything together, what is the best and what is the worst modality. And if we compare that to essentially this MDS-UPDRS measure, so this is a clinical assessment which measures how bad your motor symptoms have developed in Parkinson's disease. So it is basically, do you have disease, yes or no? So that should be a really good classification. And we can see that when we have the genetic subgroup, again, unsurprisingly, we're able to outperform actually pretty much clinical diagnosis. In idiopathic, we're not really able to outperform it. But I think the important thing that I want to show here is that irrespective of the time point and everything, and even ignoring the MDS-UPDRS, we see that in yellow, except for this little point here, is that we're actually able to improve upon a baseline model that would just predict the most accurate class, right? So the model learns something at each time point for each group, which means that there is some sort of signature or signal for Parkinson's disease in these whole blood and proteomic samples, which in itself is pretty nice because it's a lot less invasive having to give a sample of blood instead of getting a lumbar puncture for giving kind of these CSF protein samples, or for giving a brain scan, or for giving some sort of brain biopsy or something like that. So preferably, if we could do this by blood and be able to track the disease, that is much, much friendlier. And finally, I've had some feedback, particularly in paper review currently, about this figure. So I'll try and get it right this time. Basically, if you were to train a model at any time point and then test that model at that time point, you would expect that to be your best performing model. What we found is that when we actually train a model later on in the disease course and test that model earlier in the disease course, that test accuracy is higher than training that same model at year 0. So when you train at year 3 and test at year 0, you're more accurate than training at year 0 and testing at year 0. And essentially, what's cool about that is that you have the same features, you have the same signals present at year 0, but for some reason, the model isn't able to find that. Why? It's probably due to some sort of, well, it's likely inherently that these features are just too noisy and that the threshold in which you're finding isn't as conclusive or isn't as accurate as the threshold, or as clear as that threshold later on in the disease course. And I think that makes a lot of sense, because Parkinson's disease is a progressive disease. As the years go on, this signal or the disease will progressively get worse in these individuals, so these signals become clearer. And so we were actually able to, in those two signatures, which we found, we were able to backtrack that signal and outperform model trained at year 0. The other thing I want to focus on here is that for those of you who have any, or who understand probably more biology than me, what you know is that this SNP modality is essentially a fixed representation of your DNA. It is what you have at birth, and it will not change. And this one in particular, if you remember, it is from the genetic association. So the reason why you have 100% accuracy for healthy control is due to this SNP modality. It's because there's a known genetic difference between individuals who are healthy and don't have that association, and those between the prodromal early and Parkinson's disease. So that's why we have 100% accuracy there. So what's important about that means that for individuals who have this genetic mutation, or have a genetic mutation for the disease, is there is some change in this modality occurring as the disease progresses. So DNA methylation is a measure, so it's quite a slow moving measure. It's what's called kind of an epigenetic measure. So it's capturing different aspects of your environment as well as your genetics. Often most kind of like, it'd be very well known with smoking, that smoking and like alcohol would affect your DNA methylation. Not in this case, but just for example. And what's interesting is that DNA methylation changes very slowly. So personally, what we think is going on is basically the DNA methylation by year 3, that signal, because it changes slowly in individuals, it is much easier for the model to find that later in the disease course. But that same signal is actually still present at year 0. So, further work to actually see if we can track that signal back further and monitor individuals as they move from prodromal to Parkinson's disease to see how that signal changes is definitely something worth further research and work. And I think, yeah, that should be it. So in summary, untangling patient variability in Parkinson's disease is really interesting and quite a fun challenge, personally speaking. I think Parkinson's disease is something that is really, really complex, but as kind of more research and more data sets and more things come together, and by joining these different omic measures, we might be able to get a bit of a more holistic or a bit of a fuller picture on what is going on. So thanks to my supervisors, and thanks again for having me.
Okay, thank you for that. That was a really cool application of ML, I think. I have a question on, I guess, your results. Mm-hmm. So it seemed to suggest that the DNA methylation and the SNP, was it? Those were kind of the strong parameters, I guess you could say. Is that because, so just going back to Fanny Deligiani's talk, she mentioned data quality, that sort of thing. Mm-hmm. Do you think it's because the data is like less noisy, or is it biology related? Like these are genuinely the best biomarkers for Parkinson's?
Yeah, I'd love to be more of an expert on Parkinson's disease and particular DNA methylation and to nail that down on its head. I think that really kind of comes back to this sort of figure and this finding that it's better or easier to train at year 3 than it is at year 0. And essentially that 100% comes down to noise. It's the fact that when you have a population or a group of individuals who have a disease, especially a progressive disease, as you get more of them to a later stage, these biomarkers or these features become much, much easier to predict. The data quality, I would say in general, in the PPMI is pretty top notch. There's, as with any big data set, I will say the caveat, there's a lot of pain at the start, but it is worth it down the line. Yeah, in terms of looking at the kind of SNP and DNA methylation features in particular, SNPs shouldn't be noisy. they're pretty fixed, right? Your DNA, as long as the data curating process is pretty good, it should be okay because that signal isn't changing. That signal is consistent across all the time points. The fact that these DNA methylation, when you get this kind of moving population average, once you get enough of the people in that average, then you can start to get better results.
Great. Thank you very much for that. Any questions?
Thanks. So I was intrigued by the improvement by separating the genetic and idiopathic pieces. At a conference a few years ago, I've been trying to see if I could find out who this was, and I'm sorry I can't, so I can't source this comment properly, but referred to our existing ontology of disease, the taxonomy of disease that we have, which is both not very good and was invented by a bunch of historical doctors in the past as the bollocksome, which I stuck in my head as an interesting statement on the quality of our taxonomy of disease. On that basis, where things we think of as one disease is actually like lots of different etiologies, right? Could you, from your data, make a guess with some kind of unsupervised thing at how many diseases there are that we call Parkinson's?
Yeah, I think that's such fantastic and interesting point. It's so cool. So I think it really comes to this table here, right? So if I had my way and you had this genetic group, I would say that genetic group is Parkinson's disease, and then you've idiopathic, and then that idiopathic is going to be some amalgamation of everything else. It's like, I think to your point as well, historically, how did Parkinson's disease, or the label of Parkinson's disease, come about, right? And it's this idea that enough people went to their doctor with these symptoms, these motor symptoms. Exactly, right? It's trait. So it's, you have enough people with motor symptoms and then they all get lumped into this umbrella. But understanding the pathology and everything behind it is just so behind. If, for example, you're trying to do a cluster analysis, so when you, get to this stage and you have your single network and you were to separate out those individuals who are in the idiopathic group and try and cluster that and then, do the same network analysis of like how robust those clusters are and things like that. Yeah, you could, and then try and find a disease signal that way. I've got a year left. I'm moving on. and yeah, but absolutely. Do I have a question for how many Parkinson's diseases there are out there? Absolutely not. How many genes do we have? How many? I mean, especially when you go back to plot, I think, why is that not working? When you go back, I think here, right? And you see that you have all these different genetic associations going on with the disease itself. Like it's likely, because like this study as well with the genetic mutation in the genetics, it only focuses on LARC2, GBA, and SNCA. So that's the genetic associations which we've grouped in those individuals. So there are going to be other ones. There's been so many found. It's, I think doing enough to find those right subpopulations and those right groups to be able to then uniquely define it. But yeah, you're not in my lifetime, I'd say.
Yes.
Did all of your patients have CSF samples?
No, yeah, it's another.
I guess the follow-up to that would be, is there a reason that CSF came up more in the idiopathic group? Because, as in, did you know that a patient had Parkinson's before you took their gene sample, or?
Yeah, so like, big hands up in the air. I do no data curation here. It was all done like Michael J. Fox Foundation in America. Really, really good point and really, really well picked up. Right, why would the CSF not be informative in that genetic group as well? The reason is they didn't take the samples for it. They didn't do the CSF testing in those Parkinson's. They have subsequently, but they've just been doing CSF tracking in individuals who originally were prodromal and then transported on. So it's just we have this big group of individuals from, I think, their whatever arm of the cohort that I took that just didn't have CSF. They only had it in the idiopathic, but really well picked up. Yeah, it's a great point. 100%. Yeah, like as in when you go to this table here, the glaring thing that's obviously missing from this genetic group is why their CSF isn't informative, why alpha-synuclein isn't informative in them. It should be. It's just that modality is missing. As simple as that.
Um, I'll maybe ask one more question, sorry. Aside from omic data, did you think about any other, I know you didn't take the data yourself, but did you think about any other types of data? Because I think there are MRI studies,
Yeah, no, there's
elastography studies on Parkinson's brains, also non-invasive. And they find like stiffness and viscosity changes. Have you looked into that at all?
not for this application project, no. So even within the PPMI itself, I think I might have mentioned, but they have MRI scans also as well. There's a really small population of that, 100 or so. It would be a really nice thing to actually include in this as well, because the entire methodology is built around flexibility and patient inclusivity. So even if you have any overlap of patients who have genomic modality as well as having a brain image scan, we can include them. And absolutely, that would be extremely, extremely informative. For my next project application, we're moving to the GAP Alzheimer's project, and we're going to be incorporating fMRI with genomic measures. So maybe next year. Yeah.
Okay, very cool.
Okay, one last round of applause for Barry.
Cheers. Thanks.